Good afternoon, good morning, wherever you are. It's a great pleasure to welcome you to the last talk of the first set of the talks that we have in the context of the worldwide seminars on the neurobiology of addiction. And tonight, evening, we have Tommaso Patriarchi from the University of Zurich and EPFZ. And before we actually have Celine introduce him, let me already announce that we are going to continue, and we have a second set of talks that start next Thursday, so we don't make a break, next Thursday with David Belin, and then we're going to finish in June with Véronique Dochosh-Gamonet. So we have a wonderful list of speakers lined up for the next weeks. And we're already working on the program after the summer break. We definitely plan to carry this seminar series all the way to the end of the year. And I'm particularly happy for all the co-hosts that we have here. It's been really an exciting experience so far. So now, without any further ado, Celine Nicola, please introduce our today's speaker.
Hi everyone, so it's my great pleasure to introduce Tommaso Patriarchi today for the worldwide seminar series. Tommaso obtained his bachelor degree in biotechnology and his master degree in pharmaceutical biotechnology at the University of Siena in Italy. In 2015, Tommaso obtained his PhD in the same university, working on molecular mechanism of synaptic regulation by G protein coupled receptor signaling. Then between 2015 and 2019, he did his postdoctoral research at the University of California, Davis. There, he developed the first in-class genetically encoded dopamine sensor based on GPCR called dLight. This sensor enables in vivo imaging of dopamine dynamics with high spatial and temporal resolution. In September 2019, he joined the Institute of Pharmacology and Toxicology at the University of Zurich as an assistant professor for chemical neuropharmacology as a group leader. And today is going to present his work on new genetically encoded sensor to track addiction-relevant neuromodulators in vivo. So I'm happy to give you the stage, Tommaso.
Thank you very much for the nice introduction, Celine. And I also like to take a second to thank all the organizers for this wonderful seminar series, which is a true honor to be part of the invited speakers. Thank you so much. So as you have heard in my introduction, I don't really have a background in addiction. I rather develop molecular tools to study the neuromodulatory systems that are involved in neural communication, and therefore they can be very useful also and applicable to the study of addiction. Let me see. Okay. So as a note, I would like to point out also, because my lab was very recently started back in late 2019, just before the COVID pandemic started, all the recent progress done in my own lab with all the with several research projects that are ongoing in my lab are very preliminary, and therefore I thought today I would give you an overview of the recently published work that I contributed to as a postdoc in Etienne lab not long ago, and then I will conclude with considerations and future perspectives and some notes about the limitations and advantages of current tools, which I think you may find very useful. As you gather from the title slide, my work sits at the intersection between neuroscience and biotechnology or tool development. And this is particularly important point I want to make, that neuroscience, being such a complex field, necessarily draws different expertises from different fields and sits at the intersection of several different fields. Particularly in recent years, we have observed major technological developments in the fields of optogenetics, chemogenetics, and also genetically encoded tools. And these have proven revolutionary for our understanding of brain function. And I always like to give an example from a historical quote from one of the fathers of neuroscience, Ramon y Cajal. He actually wrote a nice book for junior investigators such as myself, which it's a very nice read when you start in your own lab. It gives useful advice. And if I may quote him, he wrote once that discoveries basically come out of applying new techniques to problems that have been long-standing or around for quite some time. He took this advice to his own work, and in fact, he utilized a technique that was state-of-the-art at his time in the early 1900s, which was the Golgi stain, and he could visualize for one of the first times the tiny synaptic structures on the dendrites of pyramidal neurons, which you can see in one of his drawings here. I assume he would have never even dreamt of the possibilities that just 80 years after his work, we would be able, with currently available technologies, to visualize even the activity, the functional activity of each individual synaptic spine over the stretch of dendrites in a living, awake, behaving animal. So this huge jump in technology enabled really a revolution, an optical revolution, which transformed neuroscience and led us to our conception of neuroscience as we know of it today. This particular example was made possible by genetically encoded calcium sensors, such as GCAMs. And this, the structure of which is shown on the left here. You can see that they are built based on a circularly permutated green fluorescent protein, which is basically a reconfigured green fluorescent protein in which a novel N and C terminus of the protein have been introduced inside the beta barrel, thereby opening a pore to the chromophore and leading to accessibility to the external solution. And by coupling this with a calcium binding protein calmodulin, and on the other side to a calmodulin binding peptide, one can make the fluorescence status or state of the fluorescent protein become sensitive to the external calcium concentration. These tools have been iteratively and constantly optimized over the past 20 years. The initial concept started in 1999, and as you can see, they have been several rounds of optimization over the years, which have led to exquisite levels of sensitivity for in vivo imaging. This is an example from my friend and colleague Ryan Cho when he was a graduate student in the Gradinaro lab. You can see how using GCAMP you can finally pinpoint the activity of individual neuronal subtypes within a complex circuit, even at depth within the brain, combined with green lenses and two-photon imaging. And you can also utilize light sheet microscopy, such as the work from Misha Arens at Janilia, for monitoring whole brain activity over transparent small animals, such as the zebrafish, at single neuron resolution. So really, genetically encoded calcium sensors that most of you are probably using in your research are truly transformative technology in the field of neuroscience. However, calcium, while it is a good proxy for neural activity, it is certainly not everything that's going on in the brain. There are several neurotransmitter and neuromodulatory molecules, such as monoamines, lipid-based molecules, neuropeptides, purine, and so forth. And these molecules have certainly profound effect on the firing patterns, plasticity mechanisms, gene expression profiles of the target circuits that they are released upon. And in turn, they can promote or modify and regulate various physiological and behavioral aspects, such as increasing or modulating arousal, anxiety, motivation, and they're certainly crucial in the processes underlying addiction. So it is fundamental to be able to detect and monitor the dynamics of individual neuromodulators, in addition to be able to monitor, record, and manipulate the calcium activity of neurons and other aspects of neural activity. So in order to do this, one needs appropriate tools, of course. And I would like to give you an overview first of the most state-of-the-art techniques that were available back when I entered the field in the early, around 2016 or 18. You could think of using SPECT or PET-based macromolecular microscopic imaging technologies. Of course, these methods are directly relevant for human disorders because you can apply them in humans by injecting a radio tracer directly into the bloodstream. However, they have poor temporal resolution, first of all, and spatial resolution. In fact, every pixel in these images represents the space that can enclose millions of neurons of the human brain. And these imaging modalities that don't tell us much about the modulatory properties at the microcircuitry level. In addition, they are only in an indirect measurement of changes in neuromodulatory levels, because they represent basically the accumulation of a radio tracer towards either a transporter or a receptor that it binds to. So it's an indirect measure. Alternatively, we could use analytical chemistry techniques that have been around for several decades, such as microdialysis. This involves the insertion of a a probe in a position in the brain which contains a semi-permeable membrane, which allows for the collection of extracellular fluid, including all the cocktail of molecules and neurochemicals that are present in it. And this, coupled with biochemical approaches such as MS or HPLC, allows for the unequivocal detection of this molecule and identification of these molecules. So while this approach is very sensitive and it can detect molecules even down to the picomolar range, and it can also monitor multiple molecules simultaneously, a cocktail of them, so it is also very multiplexable. However, it only provides single point measurements inside the brain, and therefore the spatial information that you can gather is rather limited. And in addition, it's also limited by slow sampling rates. In the best configuration, you can get sampling rates up to 1 minute, and therefore you cannot get second to sub-second level information on the release of various molecules. Alternatively, you could think of using fast-scan cyclic voltammetry, which relies on the oxidation potential of different neurochemicals. This involves the insertion of a carbon fiber microelectrode inside the brain. It is, once again, a single point measurement, and it has, certainly it has a very fast readout. It can go up to 10 Hertz, and it's also very sensitive because it can reveal down to 1 nanomolar levels of release, for example. However, if you're looking outside of the basal ganglia, then you will never be able to distinguish dopamine from norepinephrine, which are two very closely related monoamines, and they're released at quite similar levels outside of the basal ganglia. And so one cannot be sure using this technique what you are exactly looking at, because it doesn't have the molecular specificity that is required for this type of measurements. A few years ago, back in 2013, a first effort led to the development of a genetically encoded sensor for neurotransmitter. This was the igloo sniffer, the first of its kind, which is based on a PBP, a periplasmic binding protein derived from bacteria, which can bind glutamate like a Venus flytrap, similar to this plant. Basically clamps onto the molecule of glutamate, and the conformational change that occurs during this molecular movement imparts a large change in fluorescence, in the fluorescent state of the green fluorescent protein. So while these tools are genetically encoded, so we are capable of encoding them into an adeno-associated virus under the control of a specific promoter and express it in selected cell types. So this is a major advantage when you want to look at the release of the molecule that is detected by this tool in different cell types. And it's also fully orthogonal to the system under investigation, because certainly there are no bacterial proteins expressed in mammalians, and therefore it has less of an issue in terms of competing for endogenous proteins or signaling pathways. And these tools are also very fast, and they can be optimized relatively easier in vitro, because they can be expressed in a soluble form, and so they can be scaled up for high throughput screening approaches relatively easily. And however, they have limitations also, because there are not that many periplasmic binding protein available in nature, and they bind only a selected subset of molecules. For nowadays, we have a few, and there are not. It's very hard to rationally design them or engineer them to bind something that it's not naturally binding to. So to impart new binding specificity to these proteins is very difficult, particularly if you think of complex ligands such as neuropeptides, which are larger than monoamines and more complex in 3 dimensions. So back at the time, when the Cyclic sniffer was published, this was the best we could do, but it was still limited to a subset of neurotransmitters, and we want to push this barrier and break through these limitations. So in order to enable a more versatile approach for neurotransmitter sensor development, I thought to look at how the cells, neural cells, actually sense neurotransmitters. And they do so by leveraging on the superfamily of membrane receptors that is available in the mammalian genome of G-protein coupled receptors. These molecules comprise more than 800 members, and they bind to virtually all neurotransmitters, neuromodulators, neuropeptides that are present in the brain. So they are a very valuable pool from which to source out different scaffold molecules or proteins for sensor development. And they also, there's a lot of information out there in terms of crystallographic structures available that can provide or serve as a basis for guidance during tool development. So this is very, very useful to have. And so if you look at the overlaid structure of a recently published dopamine D2 receptor, in the inactive state is shown in gray and the active state is shown in light green, you can see how there's a critical conformational change occurring between transmembrane helix 5 and 6. And this is typically shown by crystallographic structures as a hotspot of G-protein coupled receptor activation. And because this is so conserved across class A GPCRs, it is a good candidate for insertion of a fluorescent protein for the detection of the activation of the GPCR. So that's where I started by inserting a fluorescent protein, the same CpGFP from GCAMP, into the human dopamine D1 receptor. And then I subsequently optimized the linker sequences, the amino acids linker sequences between the GPCR and the fluorescent protein through an iterative screening approach, which I will show you in the next slide. And by doing so, I was able to determine the ideal linker sequence that would maximally couple the allosteric coupling between GPCR activation and the fluorescent state of the fluorescent protein. I was able also to show that by taking the optimized fluorescent protein with optimized linkers and simply grafting it into other GPCR subtypes, we could make preliminary prototypes of biosensors. In particular, I tried a beta-1 and beta-2 adrenergic receptor, which are sensors for norepinephrine and epinephrine as well. And then I also could develop kappa and mu opioid-based sensors. Although they showed small responses, they were still valuable. And then we made a 5-HT2A based sensor and a melatonin based sensor. So these represent the starting point for further optimization, and they are indeed being currently optimized in the Tian lab. So this is the process of how I actually obtained the dLight. I introduced the green fluorescent protein flanked by 2 regions of mutagenized amino acids, and I screened in a mammalian-based assay under confocal microscope over 500 variants of this library, and I could find negatively responding variants and positively responding variants. And the largest change in fluorescence was the variant that I selected as dLight 1 for utilization. So I not only stopped there, but I also introduced additional modification on the intracellular loop 2 and on the TM5, TM6 side of the protein. And I could obtain dLight 1.2 and 1.3, which show different maximal dynamic range and different affinity levels. So I could tune it a little bit to make a few variants that could optimally suit different needs from the end user's perspective. And I could also generate a preliminary prototype that has really high affinity because it is based on a high affinity dopamine receptor, the DRD4, and this is called dLight 1.4, and it shows 4 nanomolar affinity. So it's important because these are GPCR-based sensors, and they are actually built on a human version of the GPCR. It's important to make sure that once you express them into a living animal for studying the dynamics of the ligand, that you don't alter the system too much under investigation. So, and by doing, to prove this in an in vitro system, we actually tested the expression of the dLight, compared it to a normal DRD1 receptor and or a GFP, and we looked at cyclic AMP production. In this case, you can see how the DRD1 leads to a nice dose response curve in terms of cyclic AMP production upon increasing agonist concentration, but the dLight shows similar levels as a normal GFP, so basically no cyclic AMP production in this cell line. And if it's expressed in a cell line that possesses endogenous DRD1 receptors, we do not see a shift or a significant shift in the dose response curve of the endogenous receptors, meaning that under these conditions, for example, under these in vitro conditions, we cannot observe a dramatic change in endogenous ligand sensitivity of the endogenous receptor. We also look at the internalization of the receptor to see whether it would actually internalize. And in this case, on the top, you see the fluorescence of dLight, and the bottom, you see the flag labeling to monitor total surface receptor levels. And when we actually stimulate the dLight, even for prolonged periods of time, we do not observe much internalization as compared to the DRD1, which has pronounced internalization. So it seems that this sensor does not couple well with arrestins or G proteins, which you could assume by already you could infer by the process of grafting a bulky fluorescent protein on the intracellular side of the receptor. So once the tool was optimized in vitro, we moved on to testing it ex vivo and in vivo in a progressive fashion. We expressed it via adeno-associated virus into brain slices and looked at it under two-photon microscopy. And with a rapid 0.5 millisecond electrical stimulation, we could see that we could elicit really rapid signals under two-photon microscopy with a 10 millisecond activation rate and 100 millisecond deactivation rate. And these signals could be prolonged by cocaine treatment or perfusion over the slice, which makes complete sense because we block the reuptake. And so we were expecting to see this nice prolongation of the decay. And we could also block using a specific antagonist of the DRD1, we could block the response altogether. Then we moved on to the in vivo system where we validated the dLight with optogenetics. In this case, we looked at the dLight signals in the nucleus accumbens with fiber photometry and stimulated either the dopaminergic or the GABAergic population of the VTA. And when you stimulate the dopaminergic population, you can see frequency-dependent increases in the accumbens dLight signal, which can report faithfully the frequency of stimulation up to 10 Hertz. When you stimulate GABAergic neurons, you instead see a dip in the dopaminergic tone in the accumbens, which was intriguing by itself because we did not expect to be able to see this dip. It may mean that the basal level of dLight are already partially activated by a basal tone of dopamine. So then we moved on to a classical Pavlovian conditioning task where we could pair a tone, a conditioned stimulus to an unconditioned stimulus, which is the delivery of a sucrose reward. And before the animal actually learned the association, you can see that upon presentation of the reward, there's a peak in dopamine signal, which precedes the leaking activity of the mouse. And the same can be said of the presentation of the sound, although the peak is quite small because it signals mostly novelty, but not associations with the reward. This is before the training. After the training, instead, you see a huge peak corresponding to the reward associated cue, which is the tone, and you can see the normal peak upon reward consumption. So we can track these kind of learning mechanisms at the level of dopamine release in the accumbens quite well. My colleague and friend Ali Mohebi actually used the dLight in the accumbens of rats to prove that he could actually look at a dissociation between dopamine release and dopaminergic neuron activity. In this case, you see that he could track the dLight for over 1 hour time, in a task where he would give a higher probability of rewards to the rat or a lower probability of reward shown on the top. And you can see how the basal tone of dopamine actually fluctuates and reflects the probability of release of reward consumption quite well. And even though you can see this effect on the tonic dopamine when you zoom out from the trace, if you zoom in, you still do not lose the temporal resolution. You can individually resolve individual bouts of reward consumption. It can also be used to report dopamine changes in response to addictive drug administration. This is a study from Christian Luscher, where we participated by providing the dLight. And basically, he found that actually heroin injection could lead to a significant increase in dopamine release in the accumbens, and so could cocaine. But other drugs that are not addictive, they did not show this increase. And so this could certainly provides solid support to the theories of the dopamine theories of addiction. They have been around for a while, but have always been a little bit of a matter of debate in terms of people thinking different things about this. So in my lab, we are actually currently investigating social interaction, and this is work by my postdoc Maria Chernysheva, who has started doing some photometry, and I can show you how she introduces a litter-mate same-sex mouse in the test cage, and when the animals actually approach each other, you can see that there is some dopamine activity in the nucleus accumbens. So we are further investigating this because we find it really interesting, and we want to use it also as a method for further tool validation of new tools that are in the pipeline. So the dLight is also useful not only for in couple with photometry, but also with two-photon microscopy. In this case, actually, this was done by Axel Nimmerjan and Solk. You can see how we could find a dopamine activity map at almost cellular resolution in the cortex of mice. So these mice were trained, they were sitting on a spheric treadmill, and they were trained to respond to a visual task, change in a LED display of a different shape. And they were supposed to start running when the change occurred. And if they did so, they would receive a water sucrose reward. And we could see regions of activity, which are color-coded red versus white here, which are very closely related, but they respond to functionally different phenomena. So the white regions respond to locomotion only, while the red regions respond to locomotion and reward consumption. And therefore, you can see how closely related regions have functionally distinct dopamine activity signals. And a snapshot of dopamine activity at such high resolution could not be even thinkable before the introduction of such tools. And I think this really speaks for the power of genetically encoded neurotransmitter sensors. It was also interesting to look at different layers of the cortex, and Axel found that layer 2-3 had a higher prevalence of reward-associated dopamine signals compared to layer 1. So it looks, you need to go a little bit deeper than the shallow layer 1 to see reward dopamine signals. So the next logical step for us was to develop a red-shifted version of the dopamine sensor, because no matter where you're looking at in the brain, there's never going to be a single transmitter be released. It's always a cocktail of different, there are different projections coming from all over the place, and there are many transmitters being released at once. If you look at the accumbens, for example, there's certainly dopamine, but also acetylcholine, lots of glutamate, GABA, neuropeptides, and WOTSO, and many, many more. So it is important and useful if we can develop red-shifted tools so that we can match them and mix them with green tools for multiplex investigation, so that we can look at two parameters at once. So this was the motivation for me to actually develop a red dLight. I took CPM-APPLE as a fluorescent protein, and I grafted it into the human D1 receptor once again, and performed the same screening approach. In this case, I used approximately 250 variant, and I could find one variant with a 250% response to dopamine, which you can see expressed in HEK cells at the bottom. When expressed into a mouse brain, you can see nice electrically evoked dopamine responses, once again, as with the green variant. And this can be prolonged by a reuptake blocker like GBR. and blocked by the DRD1 blocker antagonist. And it can also be co-expressed with IGLO-sniffer. And in this case, you're looking at glutamate and dopamine release events evoked by electrical stimulation. And we could actually perfuse a glutamate transporter blocker, this whey drug, and we could see that there was a significant effect on the decay kinetics of the IGLO-sniffer, but not on the dLight signals. So indicating that we are specifically looking at two different signals, and we can actually dissect them optically in the slides. Similarly to what I showed you for the green dLight, we actually did optogenetic validation of the dLight. In this case, we had channelrhodopsin expressed in the dopamine neurons of VTA, and we were recording dLight from the accumbens of rats. And we can see that it really faithfully reports different frequencies of stimulation. And Ali Mohebi, who did this work, actually also investigated the photostability of dLight over prolonged periods of time. He imaged these rats in the accumbens for over two-hour period and provided unpredicted rewards to these rats. And you can see that the amplitude of the reward-related signals does not change over the prolonged 2-hour period, and there's minimal photobleaching, indicating that this probe is really it's a really good probe for combination with photometry imaging modality. We then looked also at the activity of an axon-targeted GCAMP in dopaminergic projections in the accumbens in combination with local dopamine dynamics with the dLight. And as expected, the two activity really nicely correlate and overlap. But when you actually express the calcium sensor into the local DRD1 population of medium spiny neurons in the accumbens, and you look at dopamine together with the global activity of these neurons, you can see that the phasic dopamine reward signal is actually preceding the peak in global activity of the one spiny neurons by about 370 milliseconds. And so this really proves that we can start to ask questions in relation to the input-output relationships of various neural circuits by combining neuromodulatory probes and calcium probes. And so it was really nice to see. But we also combined two neuromodulatory probes, the IGLO-sniffer for glutamate sensing and the dLight in the accumbens. And we looked at Pavlovian conditioning once again. In the left, you see before training, and on the right, you see after training, the association of a cue to the reward. And we could see, as expected, the peak in dopamine signal corresponding to the reward consumption before training, and then corresponding to the cue and reward consumption after training. But we were certainly not expecting to see dips in glutamate signal corresponding to both cue and reward consumption. But that's what we observed, and it was super interesting to observe such dips in global glutamate dynamics. Mostly we were finding this surprising because many areas projecting to the accumbens are actually excited. Many glutamatergic projections are actually excited by cue and reward. And so we were thinking maybe we will see an increase, but we saw a decrease. And this is actually a matter of further investigation. We don't really understand what it means altogether. We also looked at, compared the SNR, the signal to noise ratio of the green dLight with the red dLight in the accumbens. And we could see that we could actually see higher signal to noise for the red probe. And one potential explanation for this is that red light is able to penetrate deeper in brain tissue because it scatters less. And therefore, perhaps we can collect light from a larger portion of the brain underlying the fiber tip. And this could be a potential explanation. And we also look at the effect on behavior in this particular instance, latency to collect the reward of the rat when there was dLight or dLight expressed in the accumbens as a proxy for either ligand buffering or affecting the natural physiology of the animal. But we did not see a striking effect in this particular case. So this brings me to the perspective and considerations part of my talk. And I would like to point out that obviously the dLights are not the only tools available out there. The group of Yulong Li has been actively developing his own tools called grab sensors, and there's nowadays a large variety of tools, both for dopamine but also for many other neuromodulators. And because of the rapid growth and exponential growth in the number and different types of tools available, together with my postdoc Marie Labouesse, I decided to put together this commentary, which came out just a few months ago, as a guide to you end users to help you navigate through this ever-growing, ever-expanding field of neuromodulatory sensors. And we hope it will be a useful reference because we make it really easy visually to identify different properties of the different tools and help you choose which ones to utilize. So if you ask me what would be one of the next steps or the hot questions that you could go after using these tools, because there's so many tools available, it's definitely interesting to look at examples of co-release, for example. Dopamine and norepinephrine are known to be co-release, or there is proof out there that they can be co-released by the locus coeruleus, particularly in the hippocampus. But definite proof or direct evidence, let's say, it's still missing. There's only indirect evidence. And combining multicolor probes for dopamine or norepinephrine could be a way to actually start addressing this question in a very definitive way and direct way. There's also evidence for dopamine glutamate co-release and many others examples of co-release in the brain. As summarized in this nice review by Trich and Sabatini. And I think it's certainly one of the fields that will benefit a lot from neuromodulatory sensors, the study of co-release mechanisms. And this could really unleash new insights and reveal new targets for the treatments of substance use disorder, because these mechanisms are partially involved and partially at play in some aspects of substance use disorder and addiction as well. And just to make a step in that direction, although we are still far away from reaching the ideal goal, I actually tested a green norepinephrine sensor in combination with the red dopamine sensor that I told you about in an in vitro cell-based assay. In this case, we have neurons expressing the dopamine, red dopamine sensor, and the astrocytes are expressing the green norepinephrine sensor. And I applied directly on top of the cells the two molecules, which you can see here, they're very closely related and they cannot be distinguished by fast scan voltammetry. And when you do that, you can see that optically, you can detect them very clearly, very specifically using these GPCR-based tools. And so while the sensitivity and the specificity levels may need to be adjusted and pushed to the limit before one can think of using this in vivo, but this at least proves the point that it's feasible and we are getting there. So that got me really excited. So if you look at, if you are a tool developer and you care about developing tools that have maximal sensitivity when using them in vivo, what it ultimately boils down to is two properties, which are dynamic range and affinity of the probes. And if you actually look at all the green dopamine sensors that are currently published and available to the public, these tend to cluster in two different groups. There's a group that has high dynamic range, up to 10-fold, approximately 10-fold increase in fluorescence with the dLight 1.3b. But this comes at the expense of affinity. These two probes actually have supramicromolar affinity. The other group or cluster has relatively lower dynamic range than the dLight 1.3b, but they can provide much higher affinity. And so it looks like there's no in between. And for whatever reason, I really can't really wrap my head around this. I don't know why, but I think I have a few ideas for pursuing approaches that can get us to the in-between, find a way to maintain the maximal dynamic range, but really increase the affinity to a level that makes significant contribution to the ultimate sensitivity when using these tools in vivo. And this is actually one of the main projects in my lab that we are currently pursuing, but it's way too preliminary for discussion. So you're all familiar with the mesolimbic dopaminergic architecture, this is a review by Eric Nestler and Christian Luscher. So you can see how there are projections from VTA to the accumbens, which are primarily involved also in the mechanism of addiction, but there are also other projections I like to keep in mind that go from the VTA to the prefrontal cortex, and these are much sparser than those that go to the basal ganglia. And if you want to look at dopamine signals in these areas of the cortex, then if you want to use the currently available dopamine sensors, you may have some troubles. The signals that you get there are not as sensitive as one would wish for. You may have to do a lot of trials, a lot of averaging, a lot of repeats, and the results may not be as robust from lab to lab. And therefore, this is one big question that I have in mind to address with new tool development. And I think that if you look at the current set of dLight sensors, what we really wish for is something with a profile that looks kind of like this. We wish to maintain maximal dynamic range, approximately 9 to 10-fold increase in fluorescence, which because it's always the best you can hope for based on the currently available probes. But we would like to engineer a way to shift the affinity and have a higher affinity probe, approximately 5 to 10 times higher affinity. I believe this would be the ideal tool to start investigating cortical and also perhaps hippocampal dopamine dynamics with a very high sensitivity in vivo. Of course, if you think of developing higher affinity probes, you have to be mindful of the side effects of this, because with high affinity comes also buffering. And we put together a graph here, me and Marie, to explain it a little bit more in detail. So if you look at, if you consider a probe with very high affinity, 2 nanomolar, when you have low dopamine levels, this probe will already be almost saturated. When you have a probe with 200 nanomolar affinity, we are much less, we have much lower levels of saturation, and even lower with a probe with 2 micromolar affinity. So depending what affinity level your probe has, the risk or the likelihood of buffering dopamine at endogenous dopamine receptors changes drastically. And there are several parameters to be considered in this, not just the affinity of the probe. The expression levels are a key parameter in this, and many other potential players in the equation that must be considered. But it's important to keep in mind that, as I said, expression levels and affinity of the sensor have a striking impact on the likelihood of buffering on the endogenous systems, and must be kept in mind when planning experiments, particularly if you're looking at behavior readout in combination with the dynamics of dopamine, because you don't want to affect or significantly change the physiology of the animals while recording the dopamine activity. And high affinity comes often at the expense of off-kinetics, because higher affinity probes have typically shows lower off-kinetics, and they may not be able to distinguish closely related dopamine activity peaks. And one example from the literature that recently came out is from the igloo-sniffer study that this group did. It's a very interesting study, and because igloo-sniffer has been around for a while, obviously the effect of buffering was first studied on this type of probe. And actually they find that while igloo-sniffer can faithfully report the vesicular release of glutamate, it also has an effect on the diffusion and uptake kinetics of glutamate itself. And it seems to compete for glutamate transporters. And because of these findings, we really need to keep our eyes open and pay attention to the buffering and potential artifacts that could come out of using these tools in living awake animals. Ultimately, one wants to have a probe with the largest possible dynamic range. Very good brightness as well helps, and keep the expression levels as low as possible, because with low expression level, you really reduce the risk of buffering. Other words of caution to keep in mind is that all these GPCR sensors that are available out there nowadays, they're all based on different subtypes of receptors, and they carry the pharmacology of those receptors. So, for example, if you were planning to use the dLight into a mouse in combination with a drug that blocks DRD1 receptors, you can't do that because you will block the dLight signals themselves. And so you have to pay attention to really the pharmacology of the receptors upon which the sensors are built when planning complex pharmacological experiments. If it's not possible to, there's no tool available for the pharmacological manipulation that you intend to do, then a good alternative would be to possibly go back to fast-scan voltammetry or microdialysis, because there are no other tools available. But we can work on engineering versions of these sensors that could be potentially insensitive to some drugs if there's such interest. And lastly, also, it's important to keep in mind that G-protein coupled receptors are complex membrane proteins, and as such, they also engage a variety of intracellular protein partners through protein-protein interactions. And a particularly understudied portion of the receptor is the C-terminus. All these G-protein coupled receptor sensors that we developed and are coming out, they are basically poorly characterized in terms of the interactions with endogenous proteins through their C-terminus. There's a variety of protein scaffolds and kinases and other protein-protein interactions that could occur, and they could in turn affect the kinetics or the stability of the sensor responses. And therefore, it's absolutely quintessential to investigate their contribution to the signals that we obtain by using these sensors. Otherwise, we run into the risk that we do a lot of imaging and we collect a lot of data, and down the road, a few years after, someone figures out that there is an interaction that actually affects sensor kinetics, and this could lead to a revisitation of the interpretation of the studies that were done. So if we want to be sure of the interpretation of the data, we need to be sure what the kinetics and the dynamics of these sensors are actually reporting. So I will stop here, and I would like to thank Lin Tian for her mentoring during my postdoc time at UC Davis, and members of her lab, and the many collaborators that made the dLight and the dLight study possible, particularly Ryan Cho, who was in the Gradinaro lab, and Ali Mohebi in the Berkeley lab. And then I would like to thank also the members of my lab, particularly the PhD students that recently joined, and the two postdocs, Marie Labouesse and Maria Chernysheva, whom you've seen are already contributing to the applicational tools in my lab. We're trying to stay half in tool development, but we are trying to also expand to end using ourselves, because we'd like to notice the difficulties of using these tools and guide our tool development by doing so. And I'd like to thank all the funding sources, particularly the ERC starting grant and the SNSF project grant, which dramatically help in building the lab and starting exciting research that we're doing right now. So with this, I can take questions if there are. I'll be happy to answer them.
Okay, Tommaso, thanks for a exceptional presentation. I really enjoyed it. There are already a few questions in the question and answer session. Please ask more. Celine is going to relay them to the speaker, and we're going to start a discussion here now.
So I'm going to start with the first one, Tommaso. So it's Oliver Barnstead. Thanks a lot for the fantastic talk. I'm going to read it. So he was wondering if you, or maybe a researcher you know, have tried to use red light for dual color two-photon imaging in vivo. If not, would you expect any potential roadblocks?
So two-photon imaging in vivo, I am not aware of who is using the dLight for that. But I've shown you some data on the two-photon imaging in slice, where we could successfully use the Agalus sniffer with the red dLight for dual color imaging of glutamate and dopamine release from the slice itself. And we also have some GCAMP and dopamine imaging data in the manuscript, which is published. I invite you to look. But yes, I think it's definitely doable. I do not foresee any particular difficulties in that, to be honest.
Because I mean, it's you are adding more and more colors, and we've been in the situation where we would like to stimulate the dopamine neurons with one fluorophore, and then look at dLight, and at the same time monitor a glutamatergic input. So we have three colors. So it's there's no limitation to these requests, no?
Yeah. So yeah, the one needs to be mindful of the spectral properties of the two probes, so that there is minimal overlap, so that you minimize the risk of bleed through, so that you are sure the signal in green is actually the dopamine signal, or the other way around. And in particular, when combining these tools with opsins, one needs to be sure that when you're imaging the tool, you're not activating the opsin, and vice versa. And for photometry, it seems to be the case, because the light powers necessary for activating opsins are much higher than those utilized for photometry imaging of dopamine probes. So you could think even of activating an opsin which has some spectral overlap with the dLight in the same area by using different light powers, or light power outputs at the fiber tip.
Okay. Celine, you want to ask the next question?
Yeah, I think it's in the same thinking way of combining two sensors together in vivo. For example, for dopamine and norepinephrine, it would depend of how much each neurotransmitter will be released. So do you have any thought on the best practice for examining sensor specificity when using two sensors together, where you cannot control the amount of the ligand that will be released?
Yeah, that's a great question. It's very central to the idea of actually monitoring co-release, or two molecules that are closely related, and they are released from the same, potentially from the same varicosity, and they can be recognized by the same receptors. I think a careful in vitro validation and careful in vitro work will be a strong support for these in vivo studies. If you can certainly co-express these tools in cell culture, in mammalian cells, and you can do titrations of dopamine or norepinephrine, for example. Let's take that as an example. And I have done that myself, and I can look at the overlap in the responses of the green or the red sensor upon titrations, fine titrations of dopamine or norepinephrine. And I can see what I call the optimal imaging window, which is a window of concentration, or a concentration range, within which you are quite safe, quite secure that you are detecting dopamine and not norepinephrine, compared to the other way around. And we are still actively trying to optimize these tools to expand the imaging window in which you are safe. You know that you are detecting one molecular species versus the other. There could be different approaches to this. We can base the sensor on different GPCR subtypes, because there are several norepinephrine receptors and several dopamine receptors, and they all have slightly different affinity levels endogenously. So one can play with the pharmacology or the GPCR subtypes and generate new tools that are more specific or more selective for norepinephrine versus dopamine. And or one can try different approaches: mutagenesis, computational modeling, and screening, basically screening. And so these are all approaches that will come together to generate the perfect pair of sensors in green and red. And I believe we will get there at some point, and we will be able to monitor selectively with absolute selectivity dopamine and norepinephrine in vivo. It's a long road and winding road, but with a lot of work, we'll get there.
Okay, another technical question that came to my mind is that most of the results you express them as delta F over F. So that is a sort of a pseudo ratiometric way of doing it. Is there any hope that we're going to get true ratiometric sensors, such that we can actually calibrate them and measure absolute values?
Yeah, that's a great question. So it is certainly possible to attach a secondary fluorophore to these sensors. We can think of a SNAP or HaloTag if you want to be using a chemogenetic approach, or you can attach a secondary fluorescent protein to the C-terminus, and this should certainly be possible and should allow you to in principle, to have a ratiometric approach for the detection of the transmitter. Of course, this comes at the expense of the spectral bandwidth, so you won't be much able to combine it with optogenetic tools, because you will have two fluorescent proteins in that case. But it is possible, and I think relatively not so difficult to do. So if there's interest, we can do that. alternative approaches could be, for quantitative imaging, could be to exploit fluorescence lifetime imaging, which actually is a more quantitative way than just looking at fluorescence intensities. Although it requires specialized equipment because you have to really measure the lifetime of those photons being emitted. And so it's a little bit more difficult to do. But there are examples in the literature, particularly by the Sabatini lab, in which they combine photometry system with single photon counting, and they achieve actually lifetime imaging of a pKa probe in combination with dLight. And that's something that it could be possible to do also with GPCR probes once they are optimized and fine-tuned specifically for lifetime.
Great. Celine?
Another question from Colin Tiggy. So have you compared the dLight signal in the nucleus accumbens between drugs of abuse and natural rewards, for example, social interaction and sucrose?
That's a great question. I think actually Christian would be more qualified to answer that for me, because I have not done that study. It was by Julie Corre in his lab. But if you look at the trace that was in the figure, it looks like the dopamine signal is quite stably going up upon the injection of those drugs of addiction, versus the reward-related dopamine signal is more phasic, and it rapidly goes down after a few seconds. So the shape looks different by eye, but I have not investigated them myself. So maybe Christian can pitch in some information.
Well, I mean, that's certainly a very important question. Now we have the tools to look at them. And one difference, as you mentioned, is the difference in kinetics. So I think this is really work in progress. And you're right, we're doing some of these questions comparing sucrose reward versus cocaine reward. This is obviously important also in the light of the previous talk by Serge Ahmed. And so, yes, I think it's really great. And having the high temporal resolution makes it also a perfect tool to look at the shift between unconditioned and conditioned stimuli. So I think it's too early to tell what the response is to the question, which is a great one. But there are many labs working on it. Absolutely. Are there more questions?
Yeah, I have
Yes?
one more question from Anna Bader. So thank you for the beautiful talk, and thank you also for mentioning the transmitter buffering issue. What are the best practice to limit this effect with the current tools? And would you recommend low viral vector titers?
Yeah, I think that's actually the most common practice for limiting buffering potential is really to reduce the titer of the virus being injected, because, as I said, a major player in the likelihood of buffering is really the expression level of the probe. If you manage to keep it as low as possible, that it's just sufficient for enabling your sensitivity, high sensitivity imaging, that's the best you can do, really. And of course, keep in mind the affinity. If you have a palette of sensors such as the dLights or the GrabDAs available, if you're looking into the accumbens, you're not necessarily, depending on the questions that you're asking, the biological question that you're after, you may need a high affinity probe or a low affinity probe. But if you use a low affinity probe, you have different likelihood of bufferings depending on the receptors, the endogenous receptors that you're looking at. There's a little bit of guidance that we put in the commentary recently published, and if there are, we are still trying to come up with a more comprehensive modeling of this buffering effect. I have to say it's a really complex question to go after because there are so many variables. You have to consider that there's not just one receptor endogenously expressed. There's 5 dopamine receptors, and each of them have different affinities being reported. And so the buffering likelihood needs to be considered at each of them. And also there's the expression of different transporters for dopamine in different brain areas, and that also must be considered. And so many other parameters, such as the viral titer, as I mentioned, but also the time of expression. If you look at 2-3 weeks after expression versus 6 months after expression of the probe, there you will have different levels of expression, because matching the viral titer, you will have constantly increasing levels of expression if you're using a synapsin promoted sensor, because it's known that the payload keeps getting expressed over a period of weeks or months under this particular promoter. So it's a very complex, multifaceted question, but I'm interested and committed to investigate it further, because I think it's central. And there are not many people are actually asking this question because it's a nasty one and difficult one. But I think it's important to be mindful and watch out for what you're actually studying, because it may lead to a slight misinterpretation of the data if you don't actually know how the tools respond and how they affect the system under investigation quite well.
Okay, so then with this note of caution to a nasty question, let's stop the session here. And thanks again for a wonderful talk and a great co-hosting by Celine Nicola.
Thank you.
Stay tuned
so much
for the next talk next Thursday, same time, 9 a.m. Pacific time, noon in New York, 6 p.m. in Geneva, and midnight in Beijing. Thank you for being with us.
Thank you very much.